In 2021, there'll be a phenomenal logistical challenge to get the vaccines to the high-risk groups within every country. And this involves potentially different cold chains, different ways to transport these vaccines, some of which will need -70 or -80 deep freezing to transport them. And we've never attempted to roll out a vaccine at the scale of billions of doses and at this speed, so this won't happen overnight.
Hi everybody, welcome back to my channel. I'm Dr. Sylvia, a health educator and general practitioner with AskAwayHealth. Are you wondering how the new COVID-19 vaccines are different from each other and from older traditional vaccines? This video will show you how the first 3 COVID-19 vaccines ever developed differ from each other. By the end, you'll understand how the new vaccines are developed different to our older traditional vaccines, what the advantages of one method are compared to another, and among the new vaccines, is one more effective than the other. Make sure you like this video, click the subscribe button in the right bottom corner of your screen under the video, and the notification bell so you know every time I publish a new video. With that said, let's find out how the new COVID-19 vaccines are different. At the time of my posting this video, we have several vaccine companies around the world developing new vaccines. In this video, I'd like to pay attention to the 3 that have already gained approval in different countries and are being administered as we speak. These are the vaccines from Pfizer, BioNTech, Moderna, and Oxford AstraZeneca. First, let's look at how the vaccines were developed. The 3 newest vaccines we have against the coronavirus infection have been developed on the back of medical technology and research going back several decades. This is known as gene-based technology, and there are 2 different categories. The first is the nucleic acid chains, and the second is the viral vector. The first group is designed using man-made messenger RNA technology. We all have natural messenger RNA in every cell of our body. They contain genetic material that instructs our bodies about creating proteins used to do different things, building up parts of our body, contributing to repairs, and so on. After decades of research, scientists can create these genetic codes in the laboratory and instruct our bodies to create specific types of proteins. In the case of COVID-19, the messenger RNA helps our bodies create the spike protein, the critical protein that allows for infection with the coronavirus. This is the technology on which the Pfizer, BioNTech, and Moderna vaccines have been developed on. The second category of gene-based vaccine is known as the viral vector or viral carrier. This is the method that has been used to develop the Oxford AstraZeneca vaccine. In the viral vector vaccine, the genetic code is transported in a weakened, less harmful virus. The common cold-causing adenovirus was used to transport the genetic material. This method is not entirely new, being similar to the way the new Ebola virus, just approved in July of 2020, was developed. So now you know the way the gene-based vaccines were developed. Secondly, how do they work? This is the next point of difference. In both cases, genetic material injected into the body is used to stimulate an immune response. In the case of the Oxford AstraZeneca vaccine, when you're inoculated, you get a piece of the SARS-CoV-2 virus sequence carried on a weakened cold-causing adenovirus. So the virus sequence is based on the SARS-2 spike protein and is bound to the DNA of the adenovirus. When the DNA enters into your cell, the virus sequence is then used to create the spike protein for the SARS-CoV-2 germ. Afterward, your body sees the spike protein, recognizes, and then mounts an immune response, giving you immunity when you face, or if you eventually face, the real virus. What about the Pfizer or Moderna vaccines? When you get your shot, you get the messenger RNA encapsulated in a lipid or fat layer that facilitates its entry to your cells. So think about the messenger RNA as a blueprint for protein development. Now, this messenger RNA contains the sequence for developing the spike protein of the SARS-CoV-2 germ. When this enters into our cells, the body receives the instruction, creates the spike protein, and then discharges the messenger RNA molecules. And this allows the body to mount an immune response, so it is primed and ready for any encounter in future with the actual virus. Essentially, these 3 vaccines work by introducing genetic information into our systems, which causes our bodies to produce specific proteins. The difference is that in the case of the Pfizer and Moderna vaccines, the genetic material is directly transferred into our cells, while the Oxford AstraZeneca vaccine uses the viral vector, that is the weakened common cold adenovirus, to deliver the genetic material. Where do you stand on these new methods for developing vaccines and medicines? Is the technology a bit too much for you? Or do you think this gives us hope for significant breakthroughs in treating challenging conditions like cancer, HIV, which we thought untreatable in the past? Let me know what you think in the comments below. The third point of difference I wanted to look at is how are these new gene-based vaccines different from the older traditional vaccines? The traditional vaccines are the ones that we've been using for decades. You know, immunization programs, travel vaccines like hepatitis, flu vaccines, or vaccines against HPV, tetanus, and hepatitis. The traditional vaccines either contain the live germ protein or the inactive germ, and so they're protein based, different from the newer ones which are gene based, and you need to have the genes delivered into your system for the protein, specific protein against the germ to be developed inside our bodies. The traditional vaccines are slower to develop compared to the newer ones. And this is simply because it's a lot more difficult to create the right protein in the lab, and it takes more time. On the other hand, the gene codes, the mRNA codes, are quicker to develop in the laboratory as soon as you know the virus sequence. And so they are easy to produce in very large quantities, although they are also unstable, like the messenger RNA, particularly the Pfizer, which has to be stored at extremely low temperatures. And lastly, is one of these new vaccines more effective than the other? And the answer is we don't know. These 3 vaccines have not been compared one against the other in terms of which is more effective in preventing infection. During the trials, they used different measures to determine how effective these vaccines, but not to compare one against the other. It might be in future there might be studies that show whether one vaccine performs better in the same sort of circumstances, but for now, we just don't know. Okay, my friends, so these are the differences between these 3 new COVID-19 vaccines that we have at the moment, as well as older traditional vaccines. Make sure you let me know in the comments section whether this answered some of your questions about the differences between the vaccine types. And if you know of any other features that make them different or similar, let me know in the comments section as well. If you like this video, make sure you give it a thumbs up. Don't forget to subscribe to the channel and click the bell so that you will know every time I publish a new video, which is usually once a week on Thursdays at 4 p.m. GMT. Stay tuned for my next video, which will be on analyzing natural birth control methods, because if you have watched the film series Bridgerton, you may understand why certain natural birth control methods are receiving a lot of attention. This is a good opportunity for us to shine the spotlight and find out just how effective and safe they are. As you wait for that video to go live, check out these 2 on here, and I will see you again soon. Stay well.